Precision oncology is a medical model that tailors a personalized therapeutic strategy according to the conditions of the individual patients. The University of Macau Faculty of Health Sciences began the Precision Oncological Research Initiative in Macau as early as 2015, with particular emphasis on the colorectal cancer, lung cancer, breast cancer, liver cancer, and the nasopharyngeal cancer. The research teams has been conducting intensive research investigating the molecular genetics, signaling pathways, clinical diagnostics, and novel therapeutic developments of cancer for years. In order to gain a deeper understanding of tumors, which is the key to its treatment, members of our faculty analyzed the initiation, progression, metastasis, and drug resistance from the perspectives of cellular level, molecular level, and the metabolomics level.
Autophagy is the process of engulfing and digesting the cell's own components, such as damaged organelle and protein aggregates. Cells use a process of autophagy to get rid of damaged parts in order to preserve its normal biological functions. As such, autophagy is closely implicated in human health and disease. In cancer, autophagy is believed to play a dual role. It can handle cancer initiation at the early stage, but promotes cancer progression at the late stage. Our research program aims to investigate how autophagy and cancer metabolism are implicated in cancer development and treatment.
Signaling at a molecular level directly dictates the function of the cell. Studying the oncogenic factors that drive the pre-malignant cells to malignant cells in tumor immunosuppressive microenvironment is very important for the prevention and precision treatment of cancer patients. Our laboratory has made several important discoveries, including the identification of PLEK-HF5 as a tumor metastasis repressor through the single-cell analysis, and S109-6L12 as oncogenic signaling that directs the reprogramming of tumor microenvironment in breast cancer metastasis development process.
Intercellular communication plays essential roles to coordinate biological functions within a group of cells. Cellular metabolites are such a group of messengers between cells. Our group utilized pluripotent stem cells as model to understand the impact of metabolism and signaling transduction on cellular functions. We review autocrine lactic acids sends feedback signals to regulate metabolism, and subsequently determines survival and cell fate. We also show that signaling lipids lead to alternative cellular states, while de novo lipogenesis does not affect pluripotency. These studies shed light on novel mechanisms of stem cell regulation and provide theoretical foundation for future technology development.
During metastasis, most tumor cells will die in circulation, and very few of circulating tumor cells will eventually form metastatic tumors. If we can understand how CTCs survive in circulatory system, we can design new therapies to reduce metastasis and prolong the life of cancer patients. To achieve this goal, we have developed a microfluidic circulatory system and use it to isolate cancer cells that could resist fluidic shear stress. We revealed that shear stress-resistant cancer cells express high levels of desmoglein-2 and plakophilin-1 proteins, which enabled these cells to form clusters and survive in circulation and further form metastatic tumors in mice. We also detected that tumor samples from lung cancer patients displayed high levels of DSC2 and PKP1 than the adjacent tissues. We are collaborating with a biotech company to develop a diagnostic kit for detecting CTCs with high metastatic abilities.
Based on the deeper understanding of tumors, the Faculty of Health Sciences has managed to achieve remarkable results in areas such as immunotherapy and nanotherapy using new materials. In addition, research teams has made advances in areas such as genetic testing on tumors, high-throughput drug screening, and clinical translational research on related technology or products, targeting the clinical treatment of tumors.
Immunotherapy has shown great promise in recent years. Unlike traditional cancer treatments, such as chemotherapy and radiation therapy, which directly target cancer cells, immunotherapy works by enhancing the body's natural immune response to cancer, making it the safest and most effective method for combating cancer. Our laboratory focuses on the identification and modification of novel human monoclonal antibodies and nanobodies targeting immunotherapy markers. We are further developing these antibodies into recombinant bispecific antibodies, or directly modifying immune cells to activate the human immune system for targeted cancer therapy. Our goal is to achieve a single multi-round attack, ultimately leading to the cure of cancer.
Developments in biomaterial science opened up the conjugation of nanotechnology and biomedical research, allowing highly effective target delivery to boost the effectiveness of cancer treatment. We are highly focused on mitophenolic network-based biomaterials, since mitophenolic network, making use of the coordination between phenolic ligands and metal ions, has emerged as the promising candidates for nanomedicine, most notably through the service as multifunctional diagnostic nano platforms. Mitophenolic network present unique properties such as rapid preparation, good biocompatibility, and pH responsiveness. Conventional chemotherapy, radiotherapy, and photodynamic therapy can all be achieved through the mitophenolic network.
The difference between individual tumors is called heterogeneity, which leads to the difficulty of developing standardized diagnostic methods. Our research mainly focuses on tumor heterogeneity. For example, how and why tumors differ in different patients with the same tumor type, and what is the difference among distinct tumor cells inside the same tumor. This provides the basis for further developments in precision treatment. Tumor heterogeneity has important implications for tumor progression, metastasis, recurrence, and drug resistance. In recent years, through bulk and single-cell level analysis of spontaneous tumors in mouse models, we have identified a few driver genes closely related to the development and progression of memory tumor, and verified their important roles in development of memory tumors through further experiments. Our research provides new thoughts and targets for the diagnosis, prognosis, and especially effective precision treatment of memory tumor.
There is a growing need for clinical translation of biomedical research to benefit the wide public. Our faculty has gained extensive experience in this aspect. For example, mesenchymal stem cells derived from the endless human embryonic stem cells via trophoblasts (TMSC) have demonstrated great promise in animal and clinical studies for the treatment of refractory disease such as multiple sclerosis, graft versus host disease, osteoarthritis, and diabetic foot ulcer. As an investigational new drug, TMSCs have been approved by the U.S. Food and Drug Administration to enter clinical trials on multiple sclerosis and were accepted for review by the China National Medical Products Administration. We also studied the tumor microenvironment using MSC.
Identification, selection, and application of cancer drugs with high efficacy are the foundation for effective cancer treatment. In order to achieve a tailor-made treatment efficacy, we have established various drug libraries, which contain hundreds of FDA-approved cancer drugs for personalized cancer therapy, and up to tens of thousands of compounds for new drug screening and identification. Using these libraries, we have established various drug-sensitive test platforms, including patient-derived organoid culture and tumor slice culture, for the purpose of quickly identify anti-cancer drugs that can be recommended to doctors for their choice. We have tested drug sensitivities on several hundreds of organoids and obtained good clinical responses and improved the quality of life of the patients. FHS continues to collaborate with hospitals, higher education institutes, and enterprises in Guangdong, Hong Kong, Macau Great Bay Area for enhancing the technological transfer and benefiting the well-being of mankind.